started. And welcome everyone. I would be very surprised if we don't have technical issues today. I just had to have my password reset and it looks like everything's running, but we will do our best and let me know if there's any issues. So here is a poll. Go ahead and it says there's 0 results. That's odd. Okay, go ahead and vote. I'll give you 10 more seconds. Or I'll give you, people are still logging in, 30 seconds. How long ago do you think bacteria became part of the human cell, so mitochondria? All right. So pretty good. 2 billion years ago to 1 trillion. So it's about 2 billion years ago. And a lot of you said, you know, knowing that rough range doesn't really matter. We're going to talk today about drugs that affect us in a negative way because of that and when we want to use them, because some of our machinery is like bacteria. So antibiotics, depending on the drug, can affect our mitochondria. Usually, obviously, we choose drugs that don't do that, that are more specific. Okay. So today is a little interesting in that we kind of always have the discussion how much to go over, and we want everyone to be on the same page. So a lot of what will be in here will be review. All the clinical vignette stuff will probably be new, and they may be some minutia, but we're just going to go over the rote machinery of how you go from DNA to protein, how the proteins are modified. I have you for a 4-hour block, so feel free to stop, ask questions anytime. Usually, I think we'll have plenty of time here, maybe get out a little early, so don't be afraid to ask questions. Okay, so transcription and translation, and there is a bunch of kind of jargon that we're going to have to get through with these acronyms just because that the proteins are named that way and do those sorts of things. I won't go through the objectives except to say in this sequence of lectures, there's maybe more number of objectives that are very discrete. So there's a lot that are just, I made them very fine, know this one thing and might have like 1 slide or 2 slides about it. So don't worry about checking them off as you go. And I will say congratulations. I know it's a big deal when you get through the first exam. It seems like it went well. So kudos to you guys. All right. So we start off with DNA. We have transcription that makes RNA, and then translation going to protein, and we'll talk about all of that in these longer lectures. And so when people have both actually good and bad, when they have disease states and things are going bad, or when they need to modify their body to make things go better, like a type 2 diabetic, kind of maybe improving their insulin sensitivity, how is that done? And there's various steps here where they can intervene, right? You can imagine intervening to transcribe DNA more. You could turn on and off RNA. You can modify protein. We'll get into all that, how you can target it in mutations. And so I'll have you for 4 hours-ish today. And then on Friday, I believe, I have you for my last synchronous section. And that's by far my favorite lecture to give you, the epigenetics lecture. And so a lot of the kind of control you'll see at the end there anyway, we'll get into that Friday. All right, so an example, and one last thing is that I'm going to, by design, hit a few things again and again just because they're a little complicated. And so here's an example where I'll talk about Tamoxifen, which is a drug used for breast cancer. And so it's confusing enough that I'll have it a few times in different slides and talks during today. So we'll start off a very high level. So Tamoxifen blocks your estrogen receptor, okay? And so the estrogen receptor normally binds to estrogen, goes in, and can cause a transcription of a bunch of genes. And Tamoxifen blocks that process, okay? Now, we'll get into the more detail about that, but it can be specific on a cell-by-cell basis dependent on what's going on in that cell, what estrogen receptor is present, and we'll get on to that. All right, so RNA compared to DNA. DNA is deoxyribose sugar, meaning this red carbon I have circled coming off does not have an oxygen, whereas the RNA does. If you look at DNA, the thymine base has this methyl group coming off. RNA does not. And so when you convert the DNA to RNA code where there's a T, it's a U. The uracil looks like the thymine except without that methyl group coming off. Obviously, DNA is double-stranded and RNA is not. But since RNA is the single-stranded molecule, it can bend back and hydrogen bond with itself, and it does that. And that structure has a lot of ramifications, which we'll talk about. Sometimes it's enzymatic activity. Sometimes it's a stability that causes transcription and changes, stuff like that. Translation changes. Okay, so here's a table of the types of RNAs. Really, you should know all of these. I'm arrowing the main ones I want you to know, and I'm underlining kind of the take-homes, okay? So mRNA is the kind of main RNA people will think about, messenger RNAs, and that RNA goes on to become a protein. But there's rRNAs, which are ribosomal RNAs, and they have a catalysis ability. tRNAs are transfer RNAs. snRNAs, small nuclear RNAs, are involved in splicing. We're going to get into all these of pre-mRNA. And then mi and siRNAs can be thought of as shutting down translation. So microRNA or small interfering RNA. And there's a difference in how they work, but we'll get into this again, but the take home here is those are currently now starting to be used as drugs. And at your level, you can just think, okay, they both shut down the RNA that they're coded for. Okay, so we've already talked about going from DNA to RNA. Now there is this double-stranded DNA, and so we have to break that apart. And so we'll get into the machinery of how that's done. And so you have to decide where you're going to break it apart. So there's a promoter region to do that. And then you need the machinery to open it and kind of take apart the energetics of the hydrogen bonding. Okay, and so we will always show you the DNA in the 5' to 3' direction if we're showing you one sequence. Okay, and I'll get into that more later. Okay, so if this is super confusing to you, you don't really have to worry about it. But there is a truth to it, so that if you imagine there's a DNA strand, which we call the template strand, right? And then from that, we're going to make, I'm talking about mRNA now, we're going to make an mRNA, okay? And so if you look at if you're looking at the mRNA in a cell and you want to know kind of what proteins are around, you oftentimes people assume the proteins is around of whatever mRNA is, okay? That's a good assumption. There's issues with it I'll talk about later, but just for ease of use, when we show you that mRNA strand, and this is kind of a kludge, we're showing you the strand that will be kind of not read off of. And so there's a DNA strand and a DNA strand, and one's copied to make an mRNA strand, right? And then the mRNA strand makes the protein. But if we show you in a DNA strand, if we show you the actual strand that's used to make the mRNA, it's the opposite, right? So like you'll read an A and then, or you'll read a G and make a C of the mRNA. And so it's getting convoluted, but I'll just show you the slide to walk through what that is. So if I'm showing you this, okay, this is 5' to 3'. The RNA made from this molecule, let me just, is synthesized in the 5' to 3' direction. And that 3' to 5' strand is not shown, but that would be the template for synthesis, okay? And so long story short, it's not just us that do this, it's anyone that would show you the DNA strand, would just make it the very easy thing to do where you're just taking the mRNA strand is the same as the DNA strand we're giving you, except you're replacing the T's with U's, okay? The only time that you wouldn't have to do this, which is kind of in the real world, is if you're in the lab and you're designing primers to go against the DNA, then you have to get the right strand to know, okay, I'm going to bind to this other DNA. So in reality, the DNA strand that was used to make this RNA down here is the other side, right? So it would be the GC. It would have a C up here for the DNA strand. Okay. That really doesn't come into play a lot, but sometimes students go down rabbit holes and get really confused about it. And so because we're just kind of cheating, everyone does, to show you the DNA strand that makes it easy to understand the RNA strand that you would get. Okay, so we're going to start off with bacteria because it's simpler, and then we'll go over to eukaryotes. So basically, bacteria are primed to make the RNA. Okay, so there's a RNA polymerase. E. coli have this RNA polymerase, and it is DNA or, and I'm asking you, do you think it's DNA or transcription factor dependent? It is DNA dependent. In our world, when we get to eukaryotes, it will also be transcription factor dependent. So if there's the DNA around accessible and these proteins, it will start making it. So, and you don't need a lot of the extra machinery that we have, because we have that extra machinery to have all this control of our DNA, right? So you don't need a primer to start synthesis. Again, you can only synthesize from 5' to 3'. Here are the subunits that are showed, and the sigma binds down to the starting region, and then here's an example of a drug that could be on the birds. Rifampicin blocks elongation. It kind of binds right there between the sigma and the beta, and that's used for tuberculosis and Legionnaires' disease and leprosy. Okay, so the sigma subunit binds, and it identifies the start sequence, the promoter region. Okay, here is a consensus sequence for the promoter region. Hold on, let me open up my chat window. Sorry, I see so I can see your things. Okay, now someone type in a C if you can answer this question, and then you'll unmute. Can someone tell me why it's a good idea to have these two consensus sequences? There's one at -10 back from the start site and one back -35. Someone type a C and I'll call on you. Normally, if we were all in a room, I'd ask for where are the engineers in the class, because I think of these as like an engineering type problem, designing software in an iPhone or something. All right, Matt, what do you got?
Would it be in case one of those is in a splice site that you could still have a promoter region?
That's possible. It's redundancy, but that's not the answer I'm looking for. So if so, for example, you could move this -35 and have it just closer, and it wouldn't be as good. It's better to have it farther away, a little bit. Joseph.
Is it possible you actually get two different functioning proteins from the same gene if you just have two different starting spots?
That's a really interesting thought. No, you're always going to start at the start codon. If I see what you're saying, that in theory there could be a start codon back here, but there's not. That's never the case. Yes, just pretty much. Let me give you a bigger example. This purple thing, imagine this purple thing stretches over both of them. It does. Okay, so the landing strip is both of them. Arthur.
I was pretty much just gonna guess. Is it just a way to ensure that the entire protein complex has kind of formed around the strand?
It is. So the thing I'm going after, I'll start pulling you guys. I appreciate you chiming in. All good thoughts. Is that it ensures directionality, right? Because they're not the same sequence. So you're about to spend all this energy and make something, and if you make it wrong, it could cause problems, and if you don't make it, it could cause problems. So imagine kind of a plane going to the back of a landing strip ready to take off. This makes sure that the plane is pointed in the right direction instead of pointed backwards where there's no runway. Okay, so this aligns it like a lock and key. This is one groove and this is another groove, and so everything's coming on and pointing in the right direction. In theory, you could do that with a small one that's not a mirror image, because that would provide directionality, but it's a lot easier if you imagine lining up a little groove of like one groove of a key and getting that exactly right, as opposed to having two triangles pointing down. You're always going to be in the right spot. So that's a generic thing you can find for proteins landing on DNA that are moving in a direction. Okay, so it can bind ahead and scan, and it gets here. And then when it gets to that section I just showed you, the -35 to -10, it becomes the closed promoter complex, okay? And when it's opening the DNA so it can start transcription, that's the open promoter complex, and then it can start going. So you can imagine if this binds and is scanning in either direction, if it scanned this way and then got to this key, the promoter section, now it knows, okay, I'm going to make the open complex and then I'm going to go in the correct direction. Okay, so once it opens up, the sigma comes off, and that's the signal that it can start going. And then this blue thing coming off is the RNA chain being made as it goes. It does take energy to do this. So there's 2 NTPs here to open up that chain. 10 base pairs of RNA are made, and after that, then the sigma gets released, and then it goes 5' to 3'. Okay. So how do we know when to stop making the RNA? The DNA codes for that. The sequence where it's going to stop is, sorry, let me back up real quick. I didn't point this out, but this sequence upstream where you're binding has a lot of T's and A's, and that's because that is a less tight hydrogen bond. G's and C's have an extra hydrogen bond, so that's a tighter thing. And so this is loose, so it's energetically easier to open up that bubble, right, to start transcription. So as you might imagine, on the flip side, when you stop, you have GC-rich regions, and so you're kind of having a harder time pulling that apart, okay? In addition, when that starts to get made, you end up with this inverted repeat, okay? And so you see this GC-GC region, then some A's and U's, so this is the RNA strand being made, and then GC-GC's, and this mirror inverted repeat means that this, like I told you, RNA can bend back on itself, forms this thing called a hairpin structure. It's called that, right? Kind of looks like that. And so that basically is a physical stop to the machinery, and so you're stopping there and you're done making your RNA. Okay. So the take-home RNAs are mRNA. Sorry, we're going to go through these RNA polymerases, okay? And so there's RNA polymerase 1, 2, and 3. 2 is the main one that makes your mRNA. So RNA polymerase 2 makes the RNA that's going to make your protein. RNA polymerase, it also makes miRNAs. RNA polymerase 3, you should think about making the tRNAs. And then RNA polymerase 1 makes the rRNAs. RNA polymerase 3 does also make one of the RNAs, the 5S. Okay. And so we're going to start talking about, we're going to get into acronymville here. So there's general transcription factors, GTFs, and those are involved in transcription for all polymerase 2 promoters. And then there's gene-specific transcription factors for individual genes, okay? So we went through easier bacterial RNA polymerases. Now we're going through eukaryotic, more complex. So we have inhibitors to these that do things. So actinomycin D is an antibiotic and anti-cancer. It is a WHO essential medicine. I think I've talked to you about this, the WHO. It has a list of like about 350 medicines that every nation in the world should have because they're so crucial. So anytime you hear one of those, it's a good thing to memorize, right? So because like I say, what should you study for boards and know as a doctor? The 350 most important drugs is not a bad thing to know. It works on prokaryotes and eukaryotes. Alpha amanitin is in death cap mushrooms, and it inhibits RNA polymerase 2. And so some of these, since the polymerases have structural similarity, they can cross-react. So for example, this also has about 1/10 as much effect on RNA polymerase 1. And it can cause liver damage. Okay, so once we have RNA, it's going to undergo splicing. And so here's the generic diagram you'll see for mRNAs. These big blocks are the exons, and the lines are the introns that get cut out. And so down here, the color-coded squares are those exons that become this one mRNA, and then not all of that sequence will be a protein, because there's this 5' UTR untranslated region and 3' UTR untranslated region. And so clearly you need RNA kind of landing strips for machinery to come down and start doing things. It's kind of like how telomerase has problems going all the way to the end. So there's these sections on the end we'll talk about that do not become amino acids. So here's the 5' end, and there's something called a 7-methylguanosine cap, and it's added to the 5' cap, or just a 5' cap. And the thing I want to point out is that the things I've pointed in circled in red here are just different. And so on a topographical level, it's kind of screaming, I'm different. And so machinery, proteins can come in and bind that and recognize that as different. Okay, so it has methyl groups coming off here. Then it has this triphosphate linker, and then it has a 7-methylguanosine moiety on the very end. Okay, and all you really need to know is that it is different and there's machinery binding to it. And that stabilizes the mRNA. And again, it aligns the mRNA of the ribosome, so for translation. Okay, so then you have your, once you have that mRNA, on the other side, you have polyadenylation. And so these A's get added to the end, and that stabilizes the mRNA, okay? So to work through this, the transcription, there's a 3' cleavage site. You don't need to know that sequence. It's 10 to 30 base pairs upstream. The GU-rich sequence downstream. And then there's this poly A tail, which can be hundreds, okay, about 200 A's long. And so this is becoming drug targetable in that if you increase the polyadenylation, you're increasing the stability of that mRNA. So if there was, a common example I'll use is P53 and MDM2. So P53 is good in that it checks your cells for, are you getting out of control? Are you maybe a cancerous cell and shutting you down and killing you? MDM2 blocks P53. So P53 is down in a lot of cancers and MDM2 is up in a lot of cancers. So if P53 was down because it was not polyadenylated, its mRNA was low and it was getting degraded, if you could stabilize or add the poly A tail to P53, to the P53 mRNA, you would make more P53 protein, okay? And so when you have this poly A tail, it increases translation, export from the nucleus. There's an example of upon egg fertilization, mRNAs get polyadenylated and kind of activated that way. Okay, this is important, but probably a little too complicated for the boards to test, but I want you to understand it, that there's this alternative polyadenylation processing, APA, okay? And so here in the blue, we have exons, okay? And then you have this long tail here. And this is kind of like the idea that was mentioned about a different promoter site. You could, and sometimes this is kind of not the normal thing, but so on the top, let's say that's your normal protein, okay? You're gonna keep this blue box 1, 2, 3, 4, and then polyadenylate there, okay? You could have a different poly A tail here, okay, and make a different protein, okay? So you could mess up the splicing and actually jump. You could just make blue, purple an end. You could skip over the purple and make blue, green, green an end. The size of this purple one can be changed. And so just know that once in a while, the processing does change the final protein product, okay? That's kind of like, you'll be told about specific cases in the clinic if there's a drug that would target that in a cancer, for example, but that's not really happening a lot now. Okay. So there's these things called P bodies, processing bodies. They're involved with mRNA quality control in the cytoplasm. And so these are kind of physical regions that you can see. They have exonucleases, decapping enzymes, microRNAs, and you're storing mRNA for future translation. So this is just kind of the physical manifestation of everything I just told you. There's this processing, polyadenylation, splicing. Where's that happen? Well, you have these P bodies that have a kind of aggregation of this stuff. Okay. So these RNA binding proteins are RBPs. They can both increase and decrease stability. And so an example is this TTP and AUF. They decrease the polymRNA of inflammatory genes. So I think I've said this before, usually when you'll be exposed to disease state or kind of pain in patients, there will often be too much inflammation. And so we're trying to lower the inflammation. And so TTP and AUF, by lowering the polyA-tails of inflammatory genes, so let's say, TNF alpha or something, you're not going to have as much inflammation, okay? And an example, in cancer, you can have increased inflammation causes increased cancer. So TTP and AUF can be down in cancer, which increases inflammation. Okay. All right. So now let's talk about the introns. They vary a lot in number in our genes. So as an example, histones have 1 exon and 0 introns, and they're about 2,000 base pairs in length. And then I think I've already mentioned to you the large gene dystrophin. It is this length, 2.4 times 10 to the 6 base pairs. It has 79 exons and 78 introns. You'll notice there's always one less intron than exon, right? Because you'll have exon, intron, exon. And so you're always going to have one less. Can someone type a C if they can tell me why it's a good idea for histones to not have, there's a few answers, correct answers to this, so better chance. Why it's a good idea for histones not to have introns and to be small, I guess. Quicker you type that C, quicker we get out. Accessibility and ease of translation. Yes, it is easier, less energy required to replicate DNA. Those are both very good answers. So you're thinking exactly right. So one, we need a ton of histones, right? So there's one thing you didn't get though. So histones we need because they're binding all our DNA. We need to make a ton of them. But the other thing is that they don't need to change, right? We don't need alternative splicing. We don't want that, right? A histone is just a widget, the cylinder widget to wrap DNA around. So you wouldn't want to alternatively splice histone. The other thing to take home is that there's some genes that we only have one copy, or one copy from mom and dad, but there's other genes like histones where we have a huge number, okay, hundreds or thousands, so this copy number variant, CNVs, and that's because you want to be able to make a ton of them very fast for something like a histone. All right, so the mechanism through which splicing happens, there is a consensus sequence for the splice points, but it's this is the minimum required thing, okay? So there's an exon here in yellow, then there's a GU at the start of the splice site. In the middle, there's an A, which is the branch point, and at the end, there's an AG. Now, clearly, this is not all that's needed because there's definitely other A's in here, but you'll see how that A becomes important, okay? And so all of them have the GU, the A, and the AG. And so what happens is from the 5' end, hold on a second. Hey, sorry. From the 5' end, there's machinery I'm going to show you that's bound. When I say machinery, I mean proteins. There's proteins bound to this RNA, and it's holding it all together as it cuts it, right? If you think of the DNA as a string, you don't want to cut it and have it float apart. So it's holding it together. It cuts the 5' end, brings it back, and covalently binds it to itself at this lariat intermediate on that A, okay? And once that's done, it can cut at the AG and bring the 2 exons together. So we're going to talk about these snRNPs, small nuclear ribonucleoprotein particles. I'll just start calling them RNPs or snRNPs. And so they're made up of in them is these snRNAs, and the snRNAs are U1, U2, U4, U5, and U6, okay? And U4 and U6 are unique in that they're both in one snRNP, okay? So let's just go through them. So you have your RNA, U1 binds, okay? Now we're just going to show you them as these letters. So when I say U1, it's one of these snRNAs, okay? After that, U2 comes in, and now you're binding the you're bending the DNA, so the RNA, so you can start interacting with different sequences, right? So here you're at the U1 binds at the GU site. U2 binds in the middle A for the lariat intermediate. Then U4 and U6, which are together in one, come with U5, and kind of everything's where it needs to be now. Then U1 and U4 come off. Splicing begins. And then U5 is a little unique in that it binds upstream of the 5' splice site. So once they're all together, then you cut off that 5' splice site, and then you have this lariat intermediate, and it comes off. You see how kind of in 2 dimensions here, you're bringing the yellow near the green, okay? You're bringing it close to form to kind of resplice it together. Okay, go ahead and answer this, and I'll give you 15 seconds. All right, for some reason, Poll Everywhere on this computer doesn't let me see how many votes there are, so I'm just going to keep going. Very good. Okay, about 20,000, right. So fruit flies have 14,000 genes, okay? And so we're not, we seem much more complex than fruit flies, but we don't have that many genes. Now I'm going to say 20,000-ish, okay, because we actually have a ton more if you think of the alternative splicing, right? So every protein, or most, not histones, but many proteins can be respliced. We'll talk about, for example, the estrogen receptor and in different tissues, that sort of thing. Okay, so alternative splicing. So alternative splicing is the joining of the exons of a gene in varied combinations, right? And so we've talked about that definition of a gene before. Multiple RNAs, and thus multiple proteins are possible through alternative splicing. Here are some examples of mRNA of alpha tropomyosin gene in different tissues. So striated muscle versus smooth muscle, fibroblasts, and brain. And so you see this one gene is making different proteins in different tissues, okay? All right, so now we're back to tamoxifen and breast cancer. So if you look at the risk of breast cancer, so the risk of being diagnosed per 100,000, in white women it's 122, in African American women it's 117. So you have this ratio which seems kind of close to each other, but when you look at specific mortality, it's much higher in African Americans. They're about 150% compared to 68%. So there's times where you'll talk about populations' predisposition to get something, but then for a slew of various reasons, genetic, socioeconomic reasons, the mortality of a population can be quite different, okay? And there are tools to kind of get at these risks. So for example, there's this Gale model screening tool you can click on, and it's a survey question, and that kind of tells you your risk over the next 5 years. Sorry, I cannot advance. Okay, so all the normal things are bad. Okay, so family history of breast cancer, prior breast cancer, alcohol, smoking. In epigenetics lecture, we'll get into shift work, which means kind of working at night or alternating hours that you work. And then these inherited mutations you've hopefully heard of, BRCA1, 2, P53, ATM, PTEN. Obesity is bad for breast cancer. Interestingly, obesity premenopausal seems to be protective. So later in life, it's worse. And postmenopausal, you have an increased 10% risk per 5 BMI that you go up. The age that a woman first menstruates, the later the better. And so girls are menstruating earlier and earlier now, which increases their risk. 0 kids is bad. So it's a 1.1 relative risk. So that means that there's a 20% increased risk of getting breast cancer if you have 0 kids versus 1 kid. And that's likely hormonal due to the pregnancy. What's good? Breastfeeding and exercise, right? Exercise is pretty much always good for everything. Okay, so what's happening with the estrogen receptor? The estrogen receptor is actually made, sorry, Solomon, you have a question? And yep, I think I caught it. Go ahead.
Yeah, I was wondering if there's anything for breast cancer in men. Are there any data to show any of this in men?
Yes, I am sure there is that data. I don't know it, and I don't have it. I can look it up, and I will post a Canvas update with that data. Okay. I just don't know that information. Good question. Okay, so here is your estrogen receptor. You have a dimer, an alpha and beta, which can combine differently. And then you have these co-regulators. You can have activators or repressors. The estrogen receptor comes in to the nucleus, binds this promoter sequence, and can cause transcription. This is not of one gene, of many genes, okay? And so there are these things called SERMs, selective estrogen receptor modulators. And so that's because they can modify estrogen different in different tissues, okay? And so here I'll have a few slides like this to kind of hammer this home in different ways. I'm going to be comparing tamoxifen and raloxifene mainly. And the take home here is when the T is tamoxifen, R is raloxifene. So R is in the blue, T is in the yellow, if you can see that. And when is it good to use these and when is it bad? So not to confuse you guys, generally, tamoxifen is used for breast cancer and raloxifene is used to prevent osteoporosis, but the data behind it is much more nuanced than that. So, for example, tamoxifen and raloxifene are both good to prevent breast cancer, okay? It's not a huge difference. There was a clinical trial that put them head to head, and tamoxifen was a little better, and so now that's what's used for breast cancer. Raloxifene can reduce endometrial cancer and osteoporosis. They're bad, but they're both bad to increase hot flashes, and you can read through those. But those aren't, normally those are not as important as kind of treating the cancer. So here's a table. If you downloaded this PowerPoint, I don't know, 48 hours ago, I had this table wrong. I have it corrected here and correct in a lecture you'll see again. But basically, here's what the organ is, breast, uterus, or bone. And here are various ligands and if they're agonists or antagonists, right? So estrogen, by definition, is an agonist of the estrogen receptor in all those tissues, okay? Tamoxifen's all of these, tamoxifen, raloxifene, and fulvestrant are all antagonists in breast. So that's all good because for breast cancer, right? You're antagonizing the estrogen receptor in breast cancer. In the uterus, just the latter two are antagonists, which is good to prevent uterine cancer. And in the bone, fulvestrant, I would expect to see more data. They're kind of studying that more recently. So the main ones right now are tamoxifen and raloxifene, but this last one is also useful. And over here, I just made a visual that I thought would kind of hammer it home based on how good and how bad they are, or how strong they are as agonists or antagonists. So on the right, going to the right in green is an agonist. And so here's an estrogen as a reference, right? It's an agonist kind of at 100% for all of them. And in the bone and in the uterus and the breast, where do these fall down? Okay. And so they're all antagonists in the breast, you see. They're split in the bone and the uterus. All right, go ahead and let's take a break. And so this same link is the same, it's going to be the same WebEx all day today for me, same link. So I will go ahead and close this so I can end the recording, and I'll start it back up. We'll meet back here at 8:50.